Okay, Husam, you can start.
Okay. Yes.
I'm sorry, Husam.
As we
Can
said,
you? This is your slideshow different presenter setting, presenter display setting. You know, can you switch your
yes.
screen so that we see the slideshow, not the presenter?
Okay. Like
Yes.
like this.
Yes.
Yes. Okay. We will discuss about this paper, which is interested papers in the artificial application on the AML, which is acute myeloid leukemia. The title of this paper it is Deep Learning Detects Acute Myeloid Leukemia and predicts NPM1 mutations status from bone marrow smear. Okay.
Which country was this paper from?
What?
Which country?
From Germany. The
From
same
Germany.
authors for the MATIC. You remember my data sets in my projects? It's the same author.
Okay.
Okay. Let we talk about first about the AML disease, which is type of leukemia. It is a blood cancer marked by overproduction of immature blood cells in the bone marrow and peripheral blood smear, which is aggressive and come with many genetic abnormalities. One of these genetic abnormalities is NPM1 mutations. NPM1, which is nucleophosmin 1, a gene responsible for the cell growth.
Hold on.
Yes.
Okay, good.
Okay. As we said before, the AML come with many genetic abnormalities. One of it is the NPM1 mutation, which is the mutation in gene of nucleophosmin 1, which is responsible, or the role of it is in cell growth and stimuli to the stress in the cells. So different AML types depend on Fab classifications show varying rates of NPM1 mutations. And the genetic abnormalities play an important role in this diagnosis of AML, with other module of diagnosis like morphological features of bone marrow smears and peripheral blood smears and flow cytometry, etc. So we have here the AML bone marrow smears, which is filled with blast cells related to the AML disease, and here we have the normal bone marrow smear, which is filled with many types of blast cells in this smear. So bone marrow, why we use bone marrow smear? Why they use bone marrow smear in this study? Because the bone marrow smears provide a more complete representation of hematopoietic and leukemia cells. So for the diagnosis of AML, we have the cytomorphological features, which is the first, or the component in the diagnosis workflow of AML. But and the genetic abnormalities also. But the cytomorphology, it is time-consuming labor and prone to human error. So we need a tool faster and more accurate. As let we say, it is alternative tool, which is AI, artificial intelligence. It is effective at handling and analyzing a huge image data sets to classify and detect AML. Why they do this studies? What's the objectives of this study? It is to develop a deep learning model to detect AML using bone marrow smear image, predict NPM1 mutation based on cell morphology, and that will give us a rapid and cost-effective screening for AML. So in this study, we talk about the methodology and begin it with the data set of this study. It's include 1,251 AML patients who have been newly diagnosed and treated with AML, and 236 for control samples from healthy bone marrow donors. So we have patients come to these centers, they're newly diagnosed with AML or treated, and control samples from the donor to the center to do this research and studies. So the data came from real-world diagnostics cases with varied morphology and staining, which will be a challenge for this model to detect and classify AML. So first of all, the slides for the patients scanned at 100 magnification, and then we will have the cell segmented and image augmented. So let we go to the study cohort, which is divided into two cohorts, patients cohorts and control cohorts. Regardless to the patients cohorts, which is, as I said before, it's 1,251 AML patients, diagnosis of AML according to the WHO classification, the age over 18, and it is available biomaterial at diagnosis. And we have in this cohort 386 with a mutated NPM1 from this cohort. The researchers exclusion the acute promyelocytic leukemia. Someone of you will ask me about why they exclude this type of AML. Because, as we know, the promyelocytic leukemia has rarely NPM1 mutation. So they will not get any cell have NPM1 mutation. For the control cohort, we have 236 healthy bone marrow from the donors, as we say before. This image of this slides will digitize and image processing and augmentation. I want to lift a note here. Here we not have a single image for control cohort and for AML patients. We have a whole slide image. So we have many cells in every slide. So if we say we augmented 5,000, more than 5,000 for AML bone marrow smear image, and augmented for above 5,000 augmented control bone marrow smear image. So we have more than a million of single cells, because every slide, the average of every slide, it is have 168 cells in every slide. So after that, the researcher split the data sets for training set and test sets, as we say here, 80% for training set to train the model in the image, and 20 for test the model, because we know the AI train in data sets and test in the other data sets. The test set, we know the AI model will not see it in the training set to confirm the accuracy of the model. So,
Husam,
Yes.
Can I ask a question now?
Yes.
When they have here, 4,163 augmented image from 1,000
Yes.
AML patients, and then
Yes.
that's for the training set, okay? And then for the
Yeah.
test set, they have almost more or less like a quarter of the training set. Um, this is just my curiosity because I saw this, training set, number of samples, trainings, test set, number of samples. Like, how is there any standard method to determine the number of you need to do the training for the model, and is there any
Yes.
standard?
Yes. You asked about the percentage of for the training set at test set, right?
Yeah, to
Yes.
determine, not to say
Yeah.
the different for the test set, but to determine that how many samples you need to train your model first in the first place. Is there any standard?
Yes. In AI application in the medical data sets, as how I can say it is as a habit or something no in all community of these studies. It is also used 80% for the training set and 20% for the test set.
So you have seen that in many papers.
Yes, yes, that's all. Okay.
I'm just wondering, if they're like in medical and health study, when it comes to the population size or the number of samples, so you have to calculate the sample size to go for the
Mhm.
to do the sampling. So
Yes.
I'm just wondering if there is any standard that's it.
I
I know
I see.
your observation
Yeah.
that is 80% they use for the training set and another 20% they use for test set. I'm just wondering if there
I
is
I ask
any standard, huh?
I asked this question for myself before and search about that. It is it is usually I get the question. It is usually do that for the medical data sets, 80% for the training and 20 for the testing set.
Okay.
So let me go for the deep learning model workflow. So we know about the deep learning. We have three steps for that. First of all, the cell segmentation using faster region CNN for the segmentation and feature extraction divided into two steps by the hematologist and they detect the range and the cell type and the characteristics like our roads and give it to the model, and the model itself do the feature extraction and extract the features like cell size and the color range automatically. And for the classification between AML and healthy donors done by the CNN and a simple neural net, let we see that more clearly here in this figure, which is illustrate the multi-step deep learning platform for this study. So first of all, we have this data sets augmented and processing on the image, and then the train faster region CNN segments the cells and detect the boundaries of the cells. So we have here human in the loop in this study, because the human here or the hematologist manual correction, do the manual correction of cell boundaries for the model. After that, we have the feature extraction here from, as we say, from the deep learning models, which is CNN, and the manual labeling cell types and characteristics by the hematologists. So we now get the cell features here and segmented it by cell of the bone marrow smears. Then we will train the deep learning model on this cell characteristics and get the results. After that, we will divide it. They will divide it the data sets to the AML classification and NPM1 status prediction. So we have AML classification. They will classify the samples as AML patients or non-AML patients. Then from the data classified by the model, they will predict the NPM1 status in these samples. So what is the results, or what is the accuracy of these models? It is good performance or not? Definitely it is good performance and robust model, which is has a segmentation accuracy 0.97, and for the classification, the accuracy was 0.91 and 0.86 for the MB mutation prediction. So the results which get from this results or from these models, we have a morphological features for the NPM1 mutation cells, which is condensed chromatin and the pre-nuclear lighting zone, which we will see after this figure. For this figure, it is represent the more performance measures of the AML classification models and NPM1 mutations. In A, we will see the ROC curve for the AML and non-AML. As we know, we will get the AURC, which is area under curve of ROC. ROC represents the receiver operating characteristic, which is tool to measure the performance of the model. If we close generally, if we close to the 1, the model has a good performance, and if we the results under that, the model has a bad performance. So here we have, for the binary classification between AML and non-AML, we have 0.969, which is 0.97 actually. So the model has good performance to classify AML and non-AML. And for B, it is represent the precision and recall. We also left a note for the precision and recall. Precision, it is the ability of the model to get the true positive and classify it correctly, and recall the sensitivity of the model to classify the true or detect the true positive here. True positive also represents the or the NPM1 mutation here. So the AUROC here, it is 0.96. For figure C here, it is represents.
Sorry, Husam, I don't understand figure B. Can you explain?
Yes, figure B, as we say, it is the precision and recall, ROC for care for the precision and recall. Precision represent the ability of the model to detect the AML patients, and recall, it is represents the sensitivity to detect the AML or classify correctly of the model for the AML and non-AML. Okay. So for the figure C, it is about the NPM1 prediction. Also, ROC curve for the true positive rate and false positive rate. I forgot that to explain what is the TBR here and what is it the FBR here. TBR, it is the true positive rate. We know the true positive, it is the we set the positive for the AML patients, and it is true positive to classify AML patients, and false positive, it is false positive to classify AML patients. How we can false positive? It is the model detects or classify the control or classify the healthy control patients as AML patients. Okay.
So when the AUC is closer to 1, it is also good for the model.
Yes, it is good performance for the model. So we observed here the model has a good performance to detect AML or to classify AML and non-AML patients, and better than the predict NPM1 mutation here. Okay. Anyone has a question in this figure?
For the false positive, the lower the better.
Right. Yes, the lower the better. Yes, for false positive, because the false positive is considered the model misclassified the sample. For the true positive rates, the higher it is better.
The ROC, can you remind me again?
Receiver operating characteristics.
Okay. So usually, I mean, I've seen in a couple of papers, usually if you have a good model, higher true positive rate and lower false positive rate, you would get something like this, right? This curve. I mean, I've observed about it. I'm just wondering, how does a bad curve look like, so that at least we can interpret it quickly. Do you have any example of that?
An example from another paper?
Yeah? No.
Now I don't have, but I read many papers use the RO.
Of course, they want to publish good paper, good results, right? Good. That's okay. You can.
Curve can be like this, but
maybe we can use that.
the number of true positive increase, the number of false positive also increase accordingly, or reverse the curve actually.
Yeah, probably. Okay, so.
Okay. Let's we see to this interested figure. After get the results, the others, they want to know why what is the interpretation of this model, why they they get this results. So now we have in A here, we have orange row here, which is represents the cells classified by the model. It is have NPM1 mutation. Okay, and they apply the heat maps here, occlusion heat maps, to detect the cells classified by the model or focused by the model. So they get here. These cells contain the consent chromatin, as we say before, and the prenuclear region. So for illustrate, I put this image from my search. It is about the blast cells, and this row indicates the prenuclear in blast cell. Okay. And for B, we have the yellow row here, which is represents the model classify the cells which not have NPM1 mutation. All right, everyone okay with this image?
Husam.
Yeah.
So in clinical setting, can we also distinguish between NPM1?
I cannot hear you clearly, Amir. Please.
Can you hear me?
Yes.
Oh, you're using my mic.
Oh yeah.
Are you using my mic?
Can you hear me?
Yes.
So I want to ask, like in clinical setting, can the diagnosis hematologist know the NPM1 mutation based on the photo, or this is just the AI can only do?
Yes. Your full question is about the hematologist if he know the characteristic of the cells has a mutation of NPM1, right?
Yes.
Yes, we say that before because it is about our roads and the color of the image and this prenuclear, as we say, this prenuclear region. So what is the model? So what's the model focus on these cells? They focus on the chromatin in the nucleus and this prenuclear cells. So also that's as how I can say, show the good performance for the model. For the combination of the opinion of the hematologist and the model.
Oh.
For this cell. Yes.
But I thought your question was different, right?
It said that hematologists can differentiate between NPM1 mutation.
Did say that?
Yeah. But
You mean by naked eyes?
Yes, by naked eye.
Are you sure of them?
Yes, doctor, because how we can confirm this diagnosis by the model if the hematologists don't know about these features, right?
For first screening, because the shape of nucleus is gonna be like different, right?
After the model finish the work, the hematologist.
No, I understand if you apply the model.
Yes.
I understand.
Yeah.
Okay. Maybe I thought the question was different. Sorry. I
That's
thought
right.
that Amir was asking if the hematologist could identify or differentiate the NPM1 mutated cells with.
Yeah, that's my question.
Okay, maybe I misunderstood everyone. Because he was explaining about the model, it's just a straight answer. They cannot.
So they only after this model
NPM1 mutation, I don't know. I'm not a hematologist.
So this is my like, because I used to internet the advanced medical center. I think like if there are something like problem with the nucleus, they will like put aside and maybe like put like, how to say, sangkaan, like
Suspect.
Suspect. Then this patient will undergo another
Test.
test.
Yes, I would agree with you. I would agree, but they cannot say like totally determine, because in hematological malignancies, there are a lot of mutations. So even though the nucleus is very condensed, you can observe it with the staining, but then you cannot conclude whether it is a NPM1 mutation or not. But
genetic testing,
yeah, they have to go through the sequencing, something like that, because it's a mutation. So I just don't know if by looking directly at this condensed nucleus here, condensed chromatin here, by the image of this microscope, they could say that this is NPM1 mutated cells, like definitely sure. So they can suspect, but they cannot be 100% sure, right?
Yeah, right.
But it also, the cells could also be mutated for something else. IDH1 mutation. What else? I don't know. In your experience, Husam, if hematologists they see the image like this, so they say
For
that
this
this is
study.
NPM1 mutation? Yes or no?
I can. So ask me about if I am a hematologist, I can classify it as NPM1 mutation or not?
Yeah.
That's the question.
If the hematologist, they see this slide, okay, they see this staining, okay, they see this
Yes.
staining, and then they say this is NPM1 mutated cell. Can they say that?
Yes, can say that if they determine the type of the cells, because the NPM1 mutations, it is found in the myeloblast cells usually.
So definitely it's NPM1 mutated cells.
Yes.
Myeloblast.
Myeloblasts, it is the higher percentage of the cells contain the NPM1 mutation.
There is no
So-
other mutation associated with myeloblasts.
I don't know about that, but for myeloblasts, it is if we have the NPM1 mutation, definitely. As they say in this paper.
Oh, okay. They say it in
Yes.
this paper.
Yes.
Okay.
Okay. So we say in a figure, it is about the cells. The model detect or classify it as NPM1 mutation cells. For B, it is blast cells. It is still for the AML patients. It is still have AML, but it is not have NPM1 mutation. Okay. So it is the different types of cell here for the AML. Let me answer Zayn here because I have a question in the chat. Okay. The 10 times oh, that's the standard. So, Doctor, you see the answer from the Zain here about the selected data of for training and test set. I never heard about that, so I want to know future. Thanks. Okay, and then asked about what the characteristics of NPM1 mutated cells that you observed. So in this paper, they observed from the model, they have the model focus on the two characteristics, which is a pre-nuclear region and the constant chromatin in the in these cells. No, don't worry. So this is the pre-nuclear, as I say before, and now we will talk about this figure in the paper. So the researchers observed that for the NPM1 mutation cells and the true positive, which is NPM1 mutation cells classified by the model, and the false negative, which is false negative represents this NPM1 mutation cells, but the model reads it as wild type mutation, wild type NPM1. What is the NPM1 wild type? It is the abnormal gene in the cells. So wild type, it is a normal NPM1, and we have the NPM1 mutation and the wild type of NPM1. So the true positive, it is classified by the model as NPM1 mutations. False negative, it is NPM1 mutation, but misclassified by the model as wild type NPM1. So in this figure, it is illustrate the variant allele frequency of NPM1 for the true positive and the false negative. First of all, we have a higher VAF for the true positives, which is mean the cells classified as NPM1 mutation. It has a higher variation or abnormalities in the genes abnormality, and the false negative, it has a lower VAF, which has a low genetic abnormalities. That's what I want to ask you, actually, Doctor, about this size of the box. We have in the small size, it represents a higher variant allele frequency, and the big size of the box here, it is about it represents a low variant allele frequency. So what is the interpretation for it? Actually, I didn't know, and I searched about that, and I didn't know the exactly answer. Can you?
No, I didn't read the paper. It's
You understand my question, right?
you don't understand.
Yeah.
What's the meaning of this?
Yes. Because the small box represent a higher variant frequency, and the big box here, it represent the low variant frequency.
I think it's from the median. So if you see there's a line in between the box. That line is the median for this graph. So if you compare those, you can see that the median is less than the median for false negative is less than true positive, and then you can see also that this line, it represents the, I think this is box plot, right? So if it's correct, then it should be the upper line means the upper range, and lower line is the lowest value. So you can see that false negative, the value are more varies compared to true positive, which is more concentrated. That's what I believe lah. But it shows that the box median and the box are higher in true positive.
So we have inverse relationship between the size of the box and the. That's what
it gives a lot of information.
Inverse relationship between the size of the box and the variant allele frequency. Right?
Oh, okay, yes, correct.
Husam, I'm sorry, I have to ask you. You don't understand about the box whisker plot, box plot here
Yes.
at all, or you don't understand the interpretation of the graph here?
No, I know the interpretation, but I want to know about the size of the box here, because if we see the true positive, which is have a higher variant allele frequency, it is the size of the box, it is small. And for the patients have a low variant frequency, it is the size of the box, it's big here. So what's the interpretation for that? Just like that. Because I don't familiar with this particular That's my question.
Like Amir said, this is the median, the right, the middle line here.
Yes.
So we just compare between these two. The true positive median line and then the false negatives. And the dots here, I think it's just the outliers. The outliers, the dot here.
Outliers.
Yes. Okay.
Triangle tu tak tahu.
Yeah, the triangle we don't know. But then in this case, usually when you have this box plot, you measure the median here. So that's what the P 0.0184, the statistical analysis, the standard deviation would determine whether it's significant or not based in between these two. So you see whether it's significant or not based on the P value, and also you compare the median here between this and that. Okay, if it's higher, then it's higher, significantly higher. In this case, it's not significant.
Yes, it is, now it is higher significant.
Yeah.
Yeah. Instead to the p value, you asked me about it in the last meeting, which is the probability of the model to detect or classify the samples by random. So if we have this this value here, the model has this value here, the model, it is highly significant. We have more P 0.05. It is random. It is not significant, statistically significant of the model. As for CI, it is confidence interval. You see, it is the for AML and non-AML, it is 0.96, CI 95%, which is between 0.9 and 0.97 for the true metric here. So it is good performance for the models. B value and CI, it is like measurements for the performance of the model or the study in general. Okay.
False negative should be lower.
Yeah.
Lower
yes.
the better.
Yeah.
The lower the better. True positive should be higher. The higher the better. In this case, the
no. False positive, it is should be lower for the better performance.
Yeah,
this is false negative. It is confused.
False negative or positive?
Yeah.
This one false negative.
False negative, it is good. It's good for the performance.
So false negative should be lower.
Yeah. No. Should be high.
False negative mean positive. Need to be lower juga lah.
No, not to be lower because it's negative, but you identify it as positive, so that's the
Yeah.
false negative.
False, yeah.
Yes.
It should be true negative, should be higher.
Yeah.
True negative,
Yeah.
true positive.
True positive should be higher, and
No.
false positive should be low.
Basically, the true one should be higher lah. The false
Yeah.
should be less.
Yeah, betul.
It's false negative also should be less. So in this case, this box plot show from the analysis of the variant allele frequency here, the true positive is higher than the false negative, which you should
Yeah.
expect, or you should anticipate from the analysis. However,
Yeah.
it's insignificant,
Mm.
not significant, because
Yes.
but what I'm trying to see here, no, not see, but they say that the model was good and everything, the performance measure, but I just don't understand how do they relate to this variant allele frequency.
What?
They showed that their model works fine, significant.
Mm.
The AUC, the ROC, they are all close to 1 here, but then when they come to this true positive and false negative of the variant allele frequency, they don't get the significant true positives and false negatives here. You got what I mean?
No, actually.
Huh? No, based on the model from figure 3, you see?
Yeah.
They have a very good model,
Yeah.
right? To predict here. Especially on the figure 3C there, you have the true positive rate and you have the false positive rate, and the
Yeah.
AUC is 0.92, which is equal to
Yes.
which is closer to 1. So
1, yeah.
the closer to 1, the better the model to predict.
Mm-hmm.
So when
No. Actually, let me illustrate this figures. This figure show how the model, how I can say, explain why the model choose these cells. Okay, for the cells of NPM1 mutation, which is classified true positive, which is NPM1 mutations, it is have a very a higher variant allele frequency. Okay, we agreement with that, right? For the model, for the cells misclassified as wild type NPM1 mutations, it is have a low variant allele frequency. So just to clear the variant allele frequency between the model classified the cells as NPM1 mutation and the model, it is NPM1 mutation but misclassified by the model. Okay, it is NPM1 mutation. All of it here for true positive and false negative, it is NPM1 mutation cells. But because the low variant allele frequency, the model misclassified it as wild type NPM1.
okay.
Okay? So it is still a good performance for the model.
Okay.
Yes, the others here illustrate why the model misclassified these cells, because the low variant allele frequency.
Understood. That's very
Yes.
clear.
Alhamdulillah. I can do that.
All right.
All right.
I'm sorry, Husam, I have to leave in 3
Yes.
minutes. Actually, I have to go to science at Bertam. Please
Yes,
continue.
okay. we will finish. Just we will take a look about the limitation and future works, just like that. About the limitations of this study, we know the complicated of AML and the connected morphology of cells in other diseases, malignant or non-malignant hematological disorders, and depend on one diagnosis model, which is the bone marrow samples. Like we have many diagnosis model like flow cytometry, cells in the peripheral blood smear, and in the needs the model for further validation and diverse global datasets from other centers in the world. So the future works it will depend on the data from different centers. They will work on the prospective data to confirm the diagnosis in the hospitals or centers, and used different modalities for diagnosis like flow cytometry, peripheral blood smears, etc., and focus on other important mutation like FLT3, parafugent, in like in acute rheumatoid leukemia. In conclusion, the deep learning offers promising AML diagnostics and supports speed up the diagnosis for AML, which is this model demonstrates the effect of the mutations on cell morphology and predicts accurately both disease and the mutation status of AML. Here. Thank you.
Okay.
I hope I can do the better today.
Yeah, well done. Yeah. Any question
Thank you very
from
much.
Intan?
I actually want to ask that false negative, but he already answered my question.
Maybe you can turn on your mic and say.
Oh,
Thank you. I can't hear anyone. No?
Can you help Intan, please?
tak ada lah.
Kena lock kat atas.
Dalam system, you could have to go to the system settings.
Will comes in.
Ada kat atas.
Can you hear me?
Yeah?
Okay, I already off my mic.
Tapi he already answered my question too.
No, just say lah. I
Oh,
wanted
okay.
to ask
Oh,
before.
I actually want to ask you about the false negative based on
Yeah.
the box and whisker plot, but you already answered my question actually.
Good. So we agree that the true positive and false negative, it is NPM1 mutation. So, yes.
Yes.
So the difference is false negative is wild type and true positive is mutated.
No, true positive and false negative, it is NPM1 mutation cells, but false negative is misclassified by the classifier as wild type NPM1 because the low variant allele frequency.
Okay. Maybe if Intan you need more clarification on this, maybe Amir can like do a quick draw on the whiteboard there. You know, Intan probably is still confused. You know, with wild type, mutated cells, and then you have this AML, and then from this AML you have different classes. You know, so
I know it before 3 or 1 week. Don't worry. About wild type.
I thought you knew it from long time ago. You are working in the pathology lab, you know. Okay. Any other question? or you guys want to continue after I leave, that's also fine.
Okay.
Actually, I want to say that
You want to
because
stay?
I have to go to my work in
Okay, yes,
Oh,
yes,
okay.
yes.
for
All
5
right.
o'clock.
Maybe we can start a bit more earlier, like 30 minutes earlier, 2:30 p.m. so
Yeah.
that you will have
Yeah,
no. Yeah.
that's good.
I was thinking to suggest today, because I have to go to
Yes.
Science at Bertam at 4. All right, Husam, thank you very much. Well done.
Thank you,
Okay.
Husam.
Thank you.
Yeah,
Doctor.
thank you.
I will text you on WhatsApp for the.
I know you sent me
papers.
text messages.
Yes.
I haven't read your messages yet. Sorry, I was fully
Don't
occupied
worry.
yesterday. I played
I know.
badminton for IPPT yesterday, and
It's
we
I know,
won
I
the
know.
third prize. And I play mixed double. So we managed to defeat the badminton coach of USM.
Because oh,
That's the, what do we call it? Not the apa? Although we lost 2-5, we won only 2 games or 2 pairs against another 5. So we lost another 5, but both of the wins were, how to say, everybody was satisfied with those 2 wins. Actually, it was very competitive.
what case IPPT?
Yes, we represented IPPT
In USM.
in the
Good luck. Good luck.
sports between the departments within USM. So the best of the best for players or athletes from this event, they will be selected and represent the university to the sports between universities.
Universities in Malaysia.
Yeah, university in Malaysia, so
Ah,
to compete.
Yeah.
Let's see.
So can you go
Good
to
luck.
No.
I don't know.
Don't know yet,
Maybe I'm not among the best or whatever, but
Top 3.
our success to defeat that in the badminton coach, it was very satisfying. Was very okay. Okay. Husam, thank you very much. Well
Thank
done.
you, thank
So
you.
keep up this kind of journal club. The way you understand the paper, you should share